Hi everyone, I'm Sichun Zhang. I'm going to present a journal that is talking about a research with a microbiome in glacier-fed stream. This article is published in this January. It's very new. First, let's learn about what is glacier-fed stream (GFS). As its name, GFS is a stream that is filled up with glacier water, and it can divide it into four types. Superglacial streams, that means the streams go on a glacier. Inglacial streams means the stream is go in the glacier. Subglacial streams means the waters go under a glacier. And proglacial streams means the stream is located downstream of glacier. And in this research, the samples are collected in proglacial streams. Why is this research important? Because GFS supply water for human populations live in mountain regions. And the microbiome in the GFS are relating to maintain the water quality for these human beings. And also, climate change are getting attention in decades. And climate change may significantly affect the microbiome in GFS, so research on the microbiome is urgent. There are several goals of the research. One is to uh, build a global and systemic study on the microbiome of GFS, because the researchers studies of GFS so far focus on regional scales. That this GFS research focus on Argentina GFS, and this one is focused on GFS in China. The second goal of the research is about biofilms, because biofilms are important for microorganisms to live in an extreme environment like GFS. And the researchers want to know how these microbiome organisms gain energy and how they successfully live in this environment. The third goal is the research want to build a foundation for further studies on how microorganisms survive under the threat of climate change. Next section is about sampling. The sediment biofilm samples are collected from 86 GFS globally, including Greenland, Andes, Norway, Alps, Uganda, Russia, Kyrgyzstan, Nepal, and New Zealand. And besides genome data, the researchers also collect environmental data, such as organic carbon, reactive phosphorus, inorganic nitrogen, and chlorophyll A content, which represent biomass of cyanobacteria and algae, and also water temperature. And these parameters are low compared to other ecosystems, which means GFS is an ultra-oligotrophic environment. That means GFS is a difficult environment for microorganisms to survive. About the metagenomic data, the research team conducts shotgun libraries and sequencing for further analysis. Biofilm samples collected from GIFs were carried on phenylchlorophyll-based DNA extraction and purification. And then the purified DNA will be processed. The process includes enzymatic uh, restriction and also PCR amplification. And then the amplified fragments, DNA fragments, will be sequenced with Illumina system. Next section is about metagenome primary analysis. The metagenomic sequence data were meant for reconstructing MACs. The full name of MACs is metagenome assembled genomes, which means microbiome genomes reconstructed from metagenome data. And there are 2,868 prokaryotic MACs total in GFS, but some MACs cannot be classified, which means there's a high level of novelty of microbiome in GFS. The prokaryotic MACs were plotted in maximum likelihood phylogenetic tree. The branch means the evolution trait and relationship between species. The color represents different classes of prokaryotes. The ring means the comparison data from other ecosystems and the abundance. We can see that there's a low overlap of MACs with ecosystems from uh, other ecosystems, such as water sample of other GFS and also samples in oceans. And this underlies both novelty and distinctiveness of the GFS sedimental prokaryotes. Among prokaryotes, archaea composition is also important. Although there's only 0.06% of the coverage, we can see that most members are from the phyla of Halobacteriota and Thermoproteota, and these archaea are involved in methanogenesis, which means they can gain energy by oxidized methane. However, whether they were released from subglacial or growing in situ in a GFS cannot be concluded yet. About eukaryotic data, there are 106 eukaryotic MACs. You can see that most members are from Chrysophyceae. Chrysophyceae belong to algae and is responsible for primary production in the GFS. Then the author compared the composition of MACs and their source. They plot in a multi-dimensional scaling ordination. We can see that the samples from the same mountains range are close, which means samples are similar when they are from the same mountain range, and nearby ranges were more similar than distant ones. The concept is straightforward. The author also compared the taxonomic composition with environmental data such as dissolved chemicals and the water temperature. The square means the correlation of environment data, and the thickness of the line means the correlation of environment data to composition. We can see that composition of MACs are highly relating to environmental parameters. Notably, several of these parameters correlate with glacier coverage, revealing glacier influence on the selective environment of GFS. Then the research team want to see if the MACs are distributed in all GFS or just in certain GFS. They use Levin index to divide MACs into generalist and specialist. Generalist means they are widely distributed, and specialistic means they distribute narrowly. We can see that most of the MACs are specialists, just live in certain GFS. The researcher further analyzed the nutrition strategy of MACs. As a result, we can see that the x-axis means different nutrition strategies, include chemoorganelles, heterotrophy, mixotrophy, and phototrophy. And the y-axis means the frequency. We can see that among specialists, uh, they include heterotroph, mixotroph, and phototroph. But among generalists, the most members are mixotroph. This may be because mixotrophy is advantageous in coping with fluctuating resource availability. Next section is about functionality of the glacier-fed stream microbiome. First, genes of enzymes involved in carbon, nitrogen, sulfur, hydrogen, and oxygen metabolism was compared with MACs. The x-axis means different genes, and y-axis means different class of bacteria. Notably, we can see that widespread occurrence of genes involved in sulfate reduction across various bacterial classes in GFS. The gene is important for bacteria to use sulfate to gain energy, which means that in GFS, most men can use sulfate as an energy source. Also, urea metabolism is prominently represented across several bacterial groups such as cyanobacteria. We've known that urea-derived nitrogen can be an energy source for nitrate reduction to ammonia for bacteria. And this may represent a lack of nitrogen in GFS and a tight interaction of cyanobacteria and other bacteria. Furthermore, 3-HP4-HP pathway is a metabolism pathway relating to utilization of inorganic carbon. We can see that most MACs can possess these genes and can utilize inorganic carbon besides organic carbon. For further analysis, the researchers classified MACs into 6 clusters based on their function of gene. And the function of gene is a reference to KO, which means KEGG or histology pathways. And we can see that each cluster may include 1 or 2 classes of bacteria. Next, the researchers want to see if solar energy effect on microbiome strategies in GFS, because in this environment, solar energy is an important source energy for bacteria. And genes relating to solar energy belongs to photosystem 1, photosystem 2, and also rhodopsin. Rhodopsin is like chlorophyll. They can receive solar energy and conduct certain functions such as ion channel or produce signal in cells. As a result, we see that x-axis means different group of genes and y means the proportion. We can see in cluster 6, the green bars, they have lots of genes in different groups such as chlorophyll, photosystem II, photosystem I. This is because cyanobacteria are in this cluster, and most of the cluster possess genes in rhodopsin, but in cluster 2, they only possess genes of proteorhodopsin. The represent the presence of rhodopsin genes without photosystems 1 or 2 is indicative of aerobic and oxygenic photosynthesis. In conclusion, the various metabolism and photosynthesis-related genes found in GFS means the microorganisms possess multiple strategies to gain nutrients. Next, the researchers want to investigate the interaction between algae and bacteria. Cyanobacteria and microalgae and bacteria will perform a symbiosis-like interaction. Algae will provide organic matter such as glucose or polysaccharide for bacteria, and in return bacteria will provide a vitamin B or siderophores for the survival of algae. First, those who want to see if bacteria possess genes encoding carbohydrate-active enzymes and sulfatase. Cellulose, fucoidan, and laminin are common polysaccharides in algae, and sulfatases are involved in polysaccharide degradation. X-axis means the different functional cluster, and Y-axis means the number of genes per meg. In cellulases, we can see each cluster possesses these genes, and clusters possess more. This may be because myxococota are in this cluster. About fucoidan, we can see that only number number 2 cluster and number 4 cluster possess this gene. And this means maybe because Bacterolia and Firmicutes are in this cluster. And about laminorenases, we can see that all the clusters possess this gene. And in sulfatase, we can also see that all the clusters possess this gene. However, the number of the genes are very few. This indicates that prokaryotes are not specialized in polysaccharide degradation in GFS, and this might be because the polysaccharides are scarce in this environment. Next, the researchers want to see genes linked to metabolism and function involved in algobacteria. According to the T-map, we can see that X-axis means a different functional cluster, and Y-axis are a different gene group. This gene group include about signal like current sensing, movement like chemotaxis and flagella, and also relating to nutrient support like vitamin B12 biosynthesis and transport, also thiamine transport and siderophores biosynthesis, and also genes relating to biofilm. The color represents the proportion of genes per meg. We can see that each cluster possesses various interaction-relating genes besides siderophores biosynthesis genes. This may be because iron is not limiting in GFS. Iron can be supplied by erosion of rocks in this environment. These results means a tight interaction between algae and bacteria in GFS. Next section, the researcher want to investigate how climate change affect the microbiome in GFS. When temperature become higher, the glaciers melt and shrink. More water go into GFS. Hence GFS become less turbid and more solar energy can be utilized by algae. Accordingly, the population of algae will increase. As previously mentioned, benzene chlorophyll A can be an indicator of the algal biomass. The researchers want to see what is the difference in GFS if benzene chlorophyll A increase. First, the researcher investigated the correlation of chlorophyll A and mixotrophy. X-axis is the amount of benzene chlorophyll A. Y-axis is the percentage of mixotrophy, which is performed heterotroph and autotroph at the same time. According to a result, we can see that when the chlorophyll A increased, the percentage of mixotroph did not show significant correlation. This indicates that mixotrophs would be abundant but invariant to benzene chlorophyll A. The researchers speculate that it's because mixotrophs possess ecological success independent of organic energy sources. Also, the researcher investigated the correlation of chlorophyll A and heterotrophy. X-axis is benzene chlorophyll A again, and Y-axis represents the percentage of chemoorganotrophy which perform anaerobic respiration. According to the result, we can see that when the chlorophyll A increased, the percentage of heterotroph also increased. This indicates that microbial energy may shift toward heterotroph due to climate change. Furthermore, the researchers analyzed the correlation of benzene chlorophyll A and genes relating to resource use in algae and bacteria, such as chemotaxis and current sensing. X-axis is the amount of benzene chlorophyll A. Y-axis represents the percentage of chemotaxis or current sensing. According to the result, we can see that when the chlorophyll A increase, the percentage of these two parameters also increase. This indicates when the climate change leads to a greener GFS, the interaction within biofilms become more complex. This phenomenon could lead to the increasing of the survival chance under climate change. Based on the research results, we can have some take-home message. According to metagenomic data, we can see that GFS microbiome is diverse and complex, comparable to other aquatic and terrestrial microbiomes. And according to the multiple metabolism pathways, we know that GFS bacteria have evolved to exploit multiple energy sources, including solar radiation, inorganic and organic resources. According to the abundance of mixotrophy, we know that mixotrophy has adapted traits of the GFS microbiome to cope with fluctuating resources and environmental conditions. And according to the various interaction and increasing heterotrophy, we found that diverse metabolic links between bacteria and cyanobacteria build the basis of the GFS food web, and they probably become stronger in the future. In conclusion, given that multiple nutrition strategy and symbiosis interaction of microorganisms interaction, the microbiome in GFS may shift in the future due to climate changes, but it will be stronger and more diverse. Here are some discussed questions about this research. First, if the GFS is going to disappear, is it important to investigate? Second, are there other environments at risk of disappearing, like GFS? Third, are there other oligotrophic environments like GFS? Fourth, what are the advantages of clustering MAGs? Because in this research, the MAGs are divided into 6 clusters, but it didn't say uh, how, why it did this. Fifth, besides genes, are there other indicators that can reflect the interactions between microorganisms? Because in this research, only genes was analyzed to represent the interaction between species. Here are references for a presentation. Thanks for your listening.